Well, good afternoon, everyone. My name is Kim Buckley, and I'm the coordinator for this afternoon's class. The Song of Ice and Fire, the 2021 Nobel Prize in Medicine for the discovery of the receptors for temperature and touch. Erika Calvo-Ochoa is an assistant professor of biology and neuroscience at Hope College. She received a BS in biomedical research and a PhD in neuroscience from the Universidad Nacional Autónoma de Mexico. And she was an NSF postdoctoral research fellow at Western Michigan University. So without further ado, I'll introduce Dr. Erika Calvo-Ochoa of Hope College.
Hi, Kim. Thank you very much, Kim, for this wonderful introduction. It's great being here just to talk a little bit about these amazing discoveries that won the Nobel Prize last year, which is amazing. As Kim briefly talked about, I teach neuroscience at Hope College. So in my neuroscience classes, I was so excited to talk about these discoveries with my students. So thank you so much for inviting me to share these discoveries with you. So yes, I was born and bred in Mexico City, a city of 30 million people. So you can imagine Holland is quite the change of pace, very chaotic city. So the only time that I feel almost like in Mexico City is during tulip time. Yeah, I got all these childhood memories of me just driving in the traffic. Science has been such a journey for me. I have been in various places throughout my career in New York City, in Kalamazoo, Michigan, and even in Japan. So I'm really happy to be settled here in West Michigan, in Holland. I joined Hope in 2020 in the middle of a pandemic. So I'm really excited about embracing Holland and embracing Hope. So for my presentation, please just be comfortable asking any questions, or if you cannot hear or understand me well, just raise your hand and I would be happy to try to accommodate your needs. So please just I'm a teacher. I love interaction with my students. You're not my students, you're members of the public. So I would love to answer your questions as we go. So really briefly, my lab, the Calvo lab, these are the current members of my lab that I hope are working really hard in SCAP as we speak. I really hope so. We study zebrafish brain regeneration. So we really don't study anything related to touch, but we love to study how the brain repairs itself because humans cannot. But zebrafish is just such an amazing model because their brains just regrow, even in adulthood. So this is the type of research that we do in my lab. And I will not talk to you much about that, but I just like to, I'm the fish lady, as they call me at the Science Center. So whenever you hear the fish lady, that's me. So what about touch? This is the issue that we will talk about today. We will talk about how touch, how embracing one another, how embracing objects and even temperatures connects us to the world, right? We're touching things all of the time. As we are sitting down, we get mechanical pressure from the chair that we are sitting on. We're touching our skin, we're touching a pen, we're sensing the air, we're sensing temperature. Sometimes we don't register that anymore because it's just so natural for most of us. But it's quite a wonderful experience to be able to experience the world through touch. So although it is quite intuitive because we're touching things all of the time, we really didn't know how touch work. So historically, touch has been perceived as a lower sense because we're such visual creatures that, touch must be not really that complicated. We're touching things all the time. So that must be quite automatic, right? We touch fire, there's a cable in our tiny brain. So this is what Descartes thought about, tiny brains. So this is it, quite simple. But is that really quite simple? Well, you will find out later today. So the five senses have been always a fascination for scientists. However, as I just told you, olfaction and touch and taste were kind of relegated to the side because vision and hearing were the most important, according to scientists. So the sense of touch is so important, but so misunderstood that it was until 1944 that a Nobel Prize in medicine and physiology was awarded to these two researchers, to Dr. Erlanger and Dr. Gasser. They described the different types of sensory nerve fibers that react to painful and not painful stimuli. So I'd like you to imagine for a second, like a time that you felt pain in your hand or in your leg or in any other part of your body. Those pain signals are felt almost automatically, and they can be quite sharp, right? We know exactly where it's hurting. So it's no wonder that pain was one of the first mechanisms that were explored by scientists. But we're talking about 1944. So these discoveries were quite historic. If you think in 1944, it's quite long ago. But still, very long ago, last century long ago. I know I tell my students last century and they just look at me like, what is this old person talking about? Like, I was born in the 80s and they feel that it was like 50 years ago. Like, not really, but well, to them. So let's talk about later 19th century, right? Which I like to feel like it was like 10 years ago. Apparently, according to my students, it was not. It was so long ago. We still didn't know how touch worked. We knew how pain and non-painful information is sent to the brain, but temperature and touch and vibrations and stretch in our skin were a complete mystery. And this is why the Nobel Prize was awarded to two researchers, because we really didn't know how our body processes this such important information. Again, we're touching things all of the time. How can we not know how that works? So then comes David Julius. Dr. Julius works in San Francisco. He worked primarily, I'm going to tell you all about it. He wasn't interested in touch. He was interested in chilies, in hot food. So he might be half Mexican and doesn't know it because Mexicans are obsessed with spicy food. I can tell you from experience. And we also have Ardem Patapoutian. Dr. Patapoutian is an immigrant from Armenia. He came to the United States in high school, fleeing the war in Lebanon. And he established himself here, became a scientist working in La Jolla, California. And he was very much interested in mechanical touch. So these two scientists didn't collaborate together. They were very good friends, but they didn't work together. And that often happens with Nobel Prizes, that they are shared among different scientists that collectively help us to understand how the world works. So we will talk about first about how we can perceive heat and temperature. And I'm going to talk about chiles a lot because this is a molecule that, well, the molecules that gives chilies their heat is a molecule that Julius was interested about. And because I'm Mexican, is slightly obsessed with it. So I'm going to ask you to imagine a lot about a time that you had a spicy food. If you're like me, that's going to be a very pleasant memory. If you don't like spicy food, that might be a little painful, but still bear with me. I like you to imagine when you have had spicy food and how that feels in the tongue, in the mouth, in the face, if it's really spicy, in the ears, perhaps. Have any of you had ear ringing when you eat very spicy food? No, it hasn't been spicy enough. It's painful. It's painful. Well, I can tell you all about it. So spicy foods, right? I guess this gentleman had ringing in his ears. It looks painful, right? So capsaicin is a chemical that gives chilies their spiciness and heat. It's a single molecule. It's a single chemical. And we can even measure how spicy chilies are. We have a scale, very scientific scale. It's called a Scoville scale that we can use based on the amount of capsaicin that chilies have. So if you're not very much fond of chilies, these are good to start. These do not approach if you are not fond of spicy chilies, right? So we can have a variety of chilies. So Julius was interested in how we can perceive these spicy, wonderful foods. So before telling you all about his discoveries, because I just don't want to tell you, sure, he discovered this protein, but I would like you to kind of come with me in this journey of how they did these experiments. So as most of you probably have heard, we have DNA in our cells, and DNA is the blueprint from our cells. It makes us who we are, makes us tall or short. In my case, it gives us wavy hair or blonde hair, or makes our skin being skin and our liver be liver, right? So it has all the information that we need. So each type of cell will have different proteins. This is a process that happens in every single cell. And these proteins are the basic molecules that fuel our cells, that make all the processes in our cells, like muscle or little molecules that convert chemicals to one to another. So to study sometimes the function of cells, we can study the DNA that is being converted into proteins. So this is what these two researchers did. They worked with DNA, with genes, with genetic information, and with the proteins that these genes made. So Julius and co-workers had a really nice hypothesis. They thought that cells that respond to capsaicin, to chili, have one gene that becomes one protein, and that this protein responds to capsaicin, right? So they said we have a specific receptor, specific molecule that allows us to perceive that heat in food. That's its only job. It's really special, and no one has heard about it, but we think that it exists. That was this hypothesis. So he tested his hypothesis. And remember, if you have any question, raise your hand. I would be happy to. Oh, yes. Yes.
They utilized South American chilies primarily, or did they go into Indonesia and India?
Oh, that's
some
a
of
really
those
good
sorts
question.
of very hot, spicy things.
So
I was
when
just
you work-
curious.
Yeah, that is a fantastic question. When you work in a lab, you can buy this capsaicin. You go to internet and you just look for all the chemicals that you want, caffeine, capsaicin, salts, and then it gets shipped to your lab in powder form. And then I imagine that he or one of his students probably tried a little bit. It was probably really bad, but you have the purified form and you can put it. Yeah, that's a fantastic question. Yes.
On this scale here, you have small numbers, very large numbers. What are the units on that scale?
It's called the Scoville units. So this is an arbitrary
Okay.
unit that was invented based on how like the spiciness.
But it's a spice hierarchy.
Yes, that's correct.
I get that. Thank you.
Yeah, so 500, 0, this is a number that it was invented. It's not like, meters or Celsius. And we go from 0 to 150,000. So again, do not approach. Do not, those really spicy chilies, you smell them and you feel the heat. It's like, oh my God. Yeah, so do not approach. Touch too, if you touch chilies, yeah, that's a really good point. And you touch your eyes, you're done for.
Was the research just done with humans? How about like deer? They have these things to keep deer away. Was any research done with animals others than humans?
This is a really good question too. So all of this research was done in animals. This is something we call basic research. It was basically just understanding how mammals can perceive spiciness. So we normally use rats. In my case, I use fish. And it is quite interesting because there are animals that cannot perceive heat, like birds. So all of those little seeds that chilies have, some birds eat them and they fly away, and there's some poop with those seeds, and voila, you have chili plants all over. If a mammal does that, that mammal is done for, right? So the ability to perceive the spiciness of chilies is only found in mammals. And somehow, some of us from hotter countries found interesting that these spicy foods were actually really good. Mexican food is like 80% chili. And I can tell you, we love the flavors, not just about the spice. But isn't that really interesting how these different animals can sense heat? Rats can for sure sense heat, but rats were not exposed to capsaicin themselves. What these researchers did was to take the sensory neurons from the rats. So they sacrificed the rats, of course, ethically and with respect, and they took those neurons from the face that we use to sense not only chili, but every food that you enjoy and eat and pain. They took those neurons out of the face and those nerves. And as I like to call it, they made a milkshake out of it. They just like put it in a blender, all the proteins, all the DNA gets exposed. So what they did is to construct something that we call DNA library. This is a quite sophisticated experiment, mostly in the 90s. So what this experiment does is that every single fragment of DNA that is being used to become a protein gets sorted out. And we can use that fragment of DNA and we can insert those fragments of DNA into cells that we grow in the lab. These cells are quite useful. They come from animals as well. And we can do all sorts of stuff to them, including this DNA we put into those cells. So we can study the function of this DNA and this protein into individual cells. So then we have all of these numbers of cells. Well, not me. In the 90s, they had all of this collection of cells, with all of these fragments of DNA. Each fragment would become a protein. So these experiments, they made a bath with all that capsaicin that they got from the chemical blender. So you can adjust the concentration, etc. And you just put it into the culture plate with the cells. And then they ask what cells are responding to this capsaicin. So those cells that responded, those cells then must have the gene and the protein that responds to capsaicin. Isn't that such an elegant experiment? It is quite hard to do, but whenever like I say it, it's like, of course, this is super easy, super elegant. It took years and years of hard work to make this happen. So then they inserted the capsaicin and they discovered a TRPB channel. And I'm going to use the word channel a lot. And I'd like to tell you what a channel is, right? So think about a cell. Think about this classroom being a cell. No one can get in or out through the walls. We need doors, right? So think about channels as gatekeepers. They are the ones, they are closed. Nothing can get either in or out. When they open, we can go in or out. But sometimes those doors are closed and we need a key, right? So channels are doors and they open when we have the key. And when they open, they allow molecules to get inside of the cells. In this case, neurons get to fire electrical impulses because neurons communicate with electricity, which is incredible. So then these channels, he discovered that the key was capsaicin. So when there was capsaicin, they were open. And this is quite interesting. I'm going to tell you a little bit more about them later. But when they open, then we're going to have an electrical response. So this is a cartoon of these channels. Capsaicin, imagine again that you're eating a very mouthful, amazing, spicy to the level of comfort that you have and you love. And you have this capsaicin floating around in your tongue. And then capsaicin finds its place in these channels, in these TRPB channels. So then, like a key and a lock, they open. They physically open. There's a gate and they open. So all of these molecules, in this case, these are ions, really tiny sodium and calcium ions. Imagine how tiny they are. They rush in, and that rushing in creates electricity. And that electricity then gets transferred to our brain. Yes.
The capsaicin molecules force open the gate?
Yes, what happens is that these proteins, these channels are very comfortable just being closed. They like it like that. So when these molecules come, they make things uncomfortable. So they have to open. It's just, it's like a chemical, how to put this? It's like a comfortable chemical way that these channels are found in. So just like a key in a lock, when you have your key and you can move it around, it works something like that. So you don't have capsaicin, they will not open because they're just comfortable like that. So capsaicin makes these channels uncomfortable. We're going to put it like that. Like chilies make people uncomfortable like that. That does work, right? Something we can get behind. So these electrical signals get transferred from your tongue to your brain immediately, right? You can tell right away if you are eating something hot, and you can know if you're going to regret that decision or not immediately. So something that this was quite not what Julius was interested about, but something he found is that these channels do even more. And again, I'm going to ask you to remember one time that you have a very spicy food, does that feel hot in the tongue? Hotter than the food that you're eating? If it's really spicy, it feels painful. And even I find in English, it's very interesting that hot can mean warm or spicy. Like in Spanish, you have one word for spicy and one word for hot temperature, and we don't use them interchangeably. But in English, hot can mean very spicy food. Why? Because these foods feel hot in the mouth, even if they're not hot. If you're eating something hot, it makes the food just taste hotter, right, in the tongue. Why does that happen? Well, these channels not only respond to capsaicin, but they also respond to hot temperatures. So whenever you touch something hot, they will open. And I like to stress how this is almost like a miracle. It's just incredible. Capsaicin is a chemical, right? It's a key in a lock. But temperature is not a chemical. Temperature is just vibration of molecules. It's not something that you can buy from a vendor. It's something that you crank up the heat. And these channels are so sophisticated that they can sense that vibrations. Yes.
Erika, just so I'm clear then, capsaicin, the mechanical or the method that it uses is chemical,
Yes.
right? To open that channel. And that is not related to the heat method, which also opens that channel.
It works in the same way.
Okay, so if I'm in Antarctica
Yes.
and it's 30 below
Yes.
and I eat a chili,
You will feel it will be hot. It will be hot.
okay,
Yeah. So maybe an experiment in February that is really, really cold outside and you eat a chili, your tongue will feel hot.
well, I know mine will. Thank you.
Now I'm going to tell you the opposite. How does it feel when you eat a mint? It's cool. And then you drink a glass of cold water and it feels even cooler, right? So we can also sense cold temperatures. So I'm going to tell you a little bit more about it. So these channels also open with hot temperatures. And they open with capsaicin too. So if you're eating something that is very hot temperature-wise and it's spicy, you're gonna feel that your mouth is burning. It's gonna be burning sensation. It's gonna be either amazing or painful. And it can be painful. That ringing in the ears that I was telling you about, that is a painful sensation that is transmitted to your nerves, because those nerves transmit pain. And these channels allows us to sense pain related to temperature. So if you touch something hot, that hot is going to feel painful because of these channels. Yes.
These cells that respond to this are in the tongue. If you touch your eyes, it can burn your eyes, right?
Yes.
But skin generally doesn't have that. So it's kind of unique to certain organs.
Yes, that's a really good question. So these temperature we can sense with pretty much all the skin and even with some organs. If you eat a very hot chili, your stomach will know it, right? And we can sense temperatures. So normally these type of cells are called mucus cells. And these are very particular, very soft lining, like inside of your nose, in your eyes, in your tongue. So normally you will not feel pain if you touch chili with your hands because of all the layers of protections, but you can definitely sense the temperature in your hands. Does that answer your question? Yes.
You used to think about the last time perhaps that you had spicy food. And when I think about it, I clearly recall the spicy aspect, but I don't really have any sense of the heat and the temperature. Is that because I'm not perhaps paying attention to that? If I pay more attention to it and the spicy aspect of it is overwhelming, or is it there's sometimes that the hot temperature doesn't relate to the spiciness?
It does relate to
Okay.
spiciness because every time you have capsaicin in there, it's gonna feel hot. So
Okay.
maybe next time you have a very
Yeah.
spicy food, just pay attention to your tongue.
Yeah, maybe I'm just again reacting so much to spice. I'm not even
Yeah.
thinking about the
But
temperature.
but we say it feels hot. It feels hot in the mouth because the temperature
Okay.
goes up a little bit.
Thank you.
Just in keeping what he said, since this is DNA driven, maybe different people with different DNA structures experience this quite differently because I don't like spicy
Yeah.
because it's always hot and burning. I don't taste
Yes.
spice.
Yes.
So there are things that we can smell and taste that are very different from different person to person, right?
Yes.
I can't remember what it is. There's a compound that you can put in someone's mouth and one person will taste bitter and someone else tastes sweet.
Yes.
It's genetically driven.
It is genetic.
So I hadn't thought about why does my husband love this stuff? And I'm just like, don't put it in my food. I'm not kidding you. Don't put it
know.
in there.
Yeah, it is genetic, and we can also train our mouths. In Mexico, we have candy that has chili on it. It's just we love the stuff. My kids, they love that, not super hot, but their tongues are used to that. Just think about when you drink coffee or tea. If you give it to a very young person, they feel it burning hot temperature wise. And we're so used to it. Like sometimes if it's not piping hot, we don't like it, right? So it's genetic, it's environmental. If you grow up loving spicy food, we're used to it. Or you might absolutely never, because it feels hot and burning, right? It feels temperature wise and even painful. Maybe you haven't paid attention to the pain. The next time, hopefully it will not happen soon. Think about the pain that burning, it can be painful. Yes.
There's some illnesses that either temporarily or permanently deprive people of sense and smell.
Yes.
One common one, COVID-19,
Yes.
can do that. When that happens, how does that affect the sensation of chili?
That's a really good question. So for tasting food, we need to have smell, right? So just think about a cold, or you had COVID and lost your sense of smell. You cannot taste, but you can still feel texture. So this will feel hot.
And you can feel temperature.
Yeah, it will feel hot temperature. Maybe you will not taste it, but because these will open, it's telling your brain, this is a hot situation. So you will feel it.
Along with this photo, why does milk work for him?
Okay, that's a really good question. Capsaicin does not dissolve in water very well. It's quite an oily molecule. So I'm gonna go back for a second. So capsaicin just finds a home in here. It's very comfortable. And because it doesn't like water very much. When we drink water like crazy, we might feel this like cool sensation, and then the hotness comes back with a revenge. Milk has fat and protein, so it washes this capsaicin away. Again, we're using chemistry to our advantage. We're washing it away. So if you're in this very uncomfortable position, eat something that is oily, some bread, even some salt, that washes that molecule away. Limes also work. Limes. Like if you have some lime and you taste some lime juice, limes, yes. Yes. I can tell you all the tricks we use. We normally don't drink milk in Mexico. We just like some salt and limes, and that helps wash away the hotness, right? Yes. I loved your questions. They're really incredible. It's the lime, yes. And we talked about being outside in the cold and feeling that burning sensation. But we can also feel cold in the mouth, right? All gum, that is this fresh spearmint or mint, pretty much every single toothpaste, it has a minty flavor. So we can feel fresh in the mouth, cool. So Julius's group and even Patapoutian's labs independently were fascinated by, okay, we know that these two channels respond to hot temperatures, but we can also feel cold temperatures. So they use menthol and mint. They discovered independently this channel that responds to menthol and that makes our mouths feel really fresh and cold. And they kept discovering other compounds in food that might activate other channels that also respond to different temperatures. These are Celsius degrees. So 60 Celsius degrees is about 140 Fahrenheit. 0 degrees Celsius is about 32 Fahrenheit. So we can tell, right? Like, is this minus 20 Fahrenheit, or is this 0 Fahrenheit, or is this maybe 32 Fahrenheit? But we are that good at knowing whether something's cold or really cold or bitterly cold that we're going to lose our fingers in a second, right? Because of these channels. And foods activate them, and foods, I just found how amazing it is that we approach these channels thanks to food, thanks to compounds in food that people have used for generations, right? In the way that we cook, and we have all these spices. It is quite amazing. And as I told you, these wide array of channels get activated from 32 Fahrenheit to about 140 Fahrenheit. So it's a whole range. And I can share with you the presentation later in case that you want to go back and tell all your friends about these channels. And now you know why mint feels so fresh and how chilies feel so not fresh. So we can talk about that. So this was Julius's discovery. Amazing. But what about touch? What about mechanical pressure? Patapoutian lab was very interested in understanding how touch is perceived. Patapoutian lab, he's a little younger than Julius, so he started his lab a little later than that, in the late 90s. So he was quite familiar with the concept that channels allows us to perceive temperature. So he wondered, maybe there is a channel that also responds to touch or to mechanical pressure. Touch is nothing but us putting some mechanical pressure in our body. But how does that happen? How can we sense whether we have a feather or a rock? We feel both, but we feel them quite different, right? We have a wide range of touch sensations. We can tell if someone is hugging us or someone is punching us. Hopefully no one will punch us, but let's just keep with the positive. If someone is embracing us or someone is just like, we can tell. So they did quite interesting experiments using something called genetic engineering. And as I told you, this comes from the genes. We have DNA that becomes a protein, and these proteins have particular functions. So what they did was that they isolated cells. These cells were, again, isolated from animals. And these cells that were isolated were cells from the skin that they know are going to respond to mechanical pressure. So they isolated these cells. They had individual cells, and they poked one cell. If you can imagine, a cell is incredibly tiny, so you need a very powerful microscope, and you need the tiniest of pipettes to just poke without bursting the cell, just slight touch. And then because cells are electric, neurons are electric, you can insert a little electrode right here and measure the electrical activity. Isn't that wonderful? I don't know, we have any electrical engineers here? No, my husband is an electrical engineer, and he's like, his mind is blown by the fact that you can measure electricity in a single cell. It's crazy, in a single neuron. So these experiments, again, I'm telling you, like, they're a piece of cake. They're actually really hard. So they had all of these cells isolated. They would poke the cell. They would see the cell response really strongly. So this cell must have a protein and a channel, perhaps, that allows the cell to respond. This group then used genetic engineering to do something that is quite wonderful. It's called a silenced gene. So we can think about genes as being turned on and off. When we have a gene that is turned on, we will have that protein. When we have a gene that is turned off, we will not have the protein. It is quite hard to silence genes or to turn them off, but we can do it in the lab. So they silenced specific genes that they predicted would encode or would make these channels, right? So they knew we had the human genome back then. So it was an exploration of all of these genes that we didn't know about, but channels have some similarities to them. So you can predict, this looks like a channel. Let's silence this gene and see what happens. They did that for hundreds and hundreds and hundreds of genes. So they found that silencing of one particular gene, just one, made the cells no longer responsive. So boom, you have the gene that makes a protein, a channel that responds to touch. So if you don't have the gene, or if your gene is defective and it doesn't work quite well, you will not respond to touch. Isn't that wonderful? They basically dived into a sea of genes and found the one. Humans have around 30,000 genes that we know, and they found the one. That is Nobel Prize worthy, right? It is incredible. They called these proteins piezo 1 and piezo 2. Piezo comes from the word pressure in Greek. So piezos 1 and 2 are also channels, like the channels for heat, but they are quite unique. There's no other channels like this. The channels for heat, imagine that they have a shape like this. They're just a cylinder, and they have a little space in the middle, and they open and close. It's just a very simple open and close. Piezo channels are just very weird. They have like gates that open like this, and they have like a lid that comes and goes. And the reason for that is that they get activated with mechanical pressure. So there's not a molecule that binds to them. It's mechanical pressure. So these channels need to be very flexible. So if our cells look like this, these channels will be closed. But when we bend, that bending is going to open them up. So they respond to bending. So when we feel a tissue like this, our channels will bend slightly, but we feel a very heavy rock, they will bend tremendously. So these channels not only allows us to feel pressure, but they have a range of the pressure we can feel. Isn't that wonderful? It's not like turning the switch on and off. It's like a rheostat, like a dimmer. So these are quite unique. So when there's pressure or mechanical shearing on the skin, they open up, they move. All of these components just give away. So what happens? This is pretty similar to what happens with TRBPs, is that ions will flow and the electrical signals will be sent to the brain because they are channels. So they're cousins. So they work in a similar way in terms of all the ions that go through, but they respond to different things. And I don't like to use the word that they are designed. They were not designed, but like evolutionary, they were selected to have these incredible features. So as we speak, as you are either touching your face or touching your arms or touching whatever you are touching, you're activating this with different degrees. And then you touch maybe your face and then you touch your pen. That change in the pressure, you're going to be able to feel that change in temperature, and it's going to be so automatic that you don't even, it's almost, you don't even feel it anymore. So these opening and closing of channels are happening millions of times per second in every single inch of your body, inside and out. Isn't that just amazing? We don't think about it. This is our brain is processing all of these signals at the same time. It is just wonderful. So, of course, we know how touch and temperature are perceived, right? So these are fundamental questions that now we know. So thanks to these two researchers. So this makes these discoveries very important just because we know. But there's more to that, because we can use this power, this knowledge to harness, to keep understanding things about physiological processes, because temperature and heat pain regulates other processes, like it allows us to regulate our body temperature, right? So if most of you are Michiganders, you're very much used to cold winters. I am not. My kids are, they go out barefoot, they don't care, and I'm always freezing because you can train. You can train to be resistant to chilly, you can be resistant to heat and to cold, right? So this ability to maintain our body temperature even during Michigan winters without dying out of freezing, that happens probably not to you, but to me, I'm thinking about the possibility of just like, this is it. This is January, this is February. So this ability to regulate our temperature comes from these channels, because these channels inform the body. It is very cold. Let's shiver a little bit more because we need to maintain heat, or less sweat because it's really hot. So we need to lose temperature. If these channels don't work, our ability to regulate our temperature doesn't work. We can cure or we can treat all sorts of pain because these channels regulate pain, right? So if we design a drug to inactivate them, we can treat pain. About touch and proprioception, we can help understand a lot of physiological processes that sometimes we don't even realize, like we don't think about they requiring touch. For example, blood pressure. We don't think about it, but blood vessels are very dynamic and they become really small or really wide based on the flow of blood that we have through them. But that regulation can change and it's mechanical. It's liquid flowing through them. So if we understand how this touch helps our blood vessels to become just wider or more thin, we can regulate blood pressure. We can understand how touch helps with our well-being and emotional regulation. In kids, for example, they are developing their sense of touch and they are so touchy. They touch everything. They're touching other people. They don't know about personal space. They're just touching. They are regulating that sense of well-being and touch and learning how to navigate the world. Their brains are wiring to understand touch. Taste is not just being very sensitive to hot foods or cold foods, but because taste relies a lot on texture, that is touch as well. So think about a food that you love that is crunchy and you're about to eat it and it's soggy, it's not crunchy anymore. Isn't that a little disappointing? That is mechanical pressure, that is not flavor. So that crunchiness or that softness of food, that texture is mechanical pressure on our tongue. So to properly taste food, we need, of course, taste, but we need mechanical pressure. We need changes in temperature. So these two types of channels also allows us to enjoy food at its fullest, because it's not just about the flavor, it's about the whole experience, right? So these were kind of amazing discoveries that we will use for decades to come, right? So these were quite amazing, quite worthy of the Nobel Prize, in my humble opinion. Amazing. And I hope that you enjoyed this talk. I would like to just give a quick acknowledgement to my lab members, which again, I hope they're really working hard. Now that I'm going to go back to the lab, I hope that they are doing all of the experiments that they love and enjoy. All of these are, of course, Hope College students, very young and talented undergrads. And we have a really generous funding from a variety of foundations and Hope College that allows us to do the research that we do. And I'd like to thank you as well for being here. And I hope that you enjoyed knowing more about these wonderful channels.
Let's give Erika a nice round of applause. Very well done. Are there any other questions? We have a bunch of them.
Yes, I love it.
We'll give Kay a turn.
A long time ago. Well, let's ask about taste buds. You're talking about cells that respond to touch. Are those the taste buds? Is that what you're talking about?
Yeah, so taste buds, imagine a blooming onion, from these restaurants that is just like the petals all over. So these taste buds are individual cells that they have receptors for chemicals like sweet, sour, etc. And they also have TRBP channels, and they also have piezo's.
Okay, and then for touch receptors, I kind of remember from a long time ago, there's little onion-shaped things in the skin.
Yes.
That would be what you're talking about. Those would not be attached.
So those are like little organs beneath the skin, and we have 5 different organs. Some of them allows us to perceive just like very intense touch or intense pressure or very light pressure. Stretch, like if you stretch your fingers, it feels different or more painful. All of them are like larger organs that have different types of cells, and these cells all have piezo's on them. But they have it in a different way. So we can feel, not only feel, but we can feel, is this stretchy or is this pushing or is this more like light? So it's very specialized.
You talked about tinnitus. I never knew the connection between chili and tinnitus, but I've suffered tinnitus for almost 50 years. And I grew up without Latino food, so I can't blame it on chili. But I've seen ads for CBD to
Mhm.
treat tinnitus. Do you know anything about that?
Well, CBD has a lot of uses. And CBD, or the CBD is a compound from cannabis, and THC, which has all the other psychoactive effects. So these also activate this, but CBD also CBD also inhibits pain receptors. So that's quite interesting because it's an interplay, and it also affects receptors in the brain, pain receptors in the brain. So it's quite complicated. So what happens in the ear is that we have the sensory nerve that allows us to hear, but we also have pain sensory. So think about when you have like ear infection and it feels really bad. And this is connected to the nose and it's connected to the throat as well. So all of these nerves are, it's just wonderful. It's like a network of sensory, and we also use nerves to move our face. So chili, if it's spicy enough, yeah, you will feel it with your whole face. Yes, believe me, you will feel it with your whole face. Yes.
I mentioned that I didn't feel warmth from the spicy food, and this might be just me dreaming, but in the lab, is there anybody researching perhaps a Michigan winter diet where you could eat certain foods that would impact on feeling cold in the winter?
I don't know, that sounds very interesting.
I mean, I might need to eat spicier food more often. Would that help?
That I particularly love. Well, I'm gonna ask you, for example, if you drink hot cocoa, right? We put cinnamon on it, but mostly in the fall, or the Thanksgiving pies, they're full of spice because that is warm. Not hot, I want to die hot, but cinnamon adds that spice and your tongue will feel hotter. It's going to be slightly hotter.
I have a question about the molecular biology of this. I think the first receptors were discovered were the beta adrenergic receptors, really sorted out in terms of structure and so forth.
Yes.
And in the olfactory receptors, I think there are just many, hundreds or thousands of different ones, and which are related to each other.
Mhm.
Are these, the piezoreceptor seems to be, the picture seems to be different from others,
Yes.
but are the pressure receptors and the capsaicin receptors, are they homologous with the others? Do you know? Have
They
the
are
structures
not.
been solved?
So beta adrenergic receptors, they respond to adrenaline, and they were one of the first ones to be discovered. They are something we called GPCRs. They're like a barrel, and they have proteins inside that move and activate a bunch of proteins inside of the cell. Olfactory receptors are also GPCRs, and they bind molecules that we can perceive. And these are channels, so these are more like a gate. And piezos are quite unique. There's nothing like them.
Has the structure of these piezoreceptors really been sorted out in terms of like X-ray crystallography?
Yeah, exactly. So Patapoutian lab did that. So they discovered these receptors and they did the crystallography. So we know that, I'm showing this cartoon, but scientists have seen this in real life. They know how this looks like. They know how all of these components move with mechanical pressure.
So Erika, that to me feels like a pretty analog mechanism in the piezo. compared to a chemical
Yes.
on off kind of.
Yeah,
thing.
because it's stretching, it's
Yes.
moving.
And it's by degree.
Yeah, and
Yeah.
TBRP, I'm not gonna say it's simple, but it's just open, close, and this is like very open or slightly open, so it's more sophisticated, it has more moving parts, and then it comes back, almost like a spring of sorts, you know, you can like move it around, so it's quite sophisticated. Yeah.
I have a question. As a child, I was given a meal of liver. And immediately my throat kind of closed up on me. And the consistency or something of
Oh my
of it
God.
did that to me. And you hate to say, well, I can't eat it. But it didn't want to go down just
Yeah.
because something in me did that. Would that be caused by some sensory thing like this?
Oh yeah, absolutely. And it's also psychological, right? Sometimes, like I accidentally had brains once in tacos. This sounds very bad, but it was accidental. I didn't know. And they were quite tasty. And when someone told me what they were, I could not touch them. I gagged. All psychological. They were very good. I would never taste them again. I work with them. I don't want them in my plate. So that texture, that flavor, or that this is a liver, you know, it looks kind of funky. So it's psychological and it's physiological. It's taste and food are just incredible. Yes.
An early career of mine was 6 years as a nuclear propulsion engineer. And we learned through pictures like this. And I'm trying to put a size scale on this cartoon. Would that be 10 angstroms?
This is about, so this is the cell membrane. The cell membrane is like the walls in this room. This is about 3 nanometers.
Okay.
So a nanometer is, it's really impossible to convert that to feet, I don't think, in imperial system. It doesn't make sense. Yeah.
Angstrom is, I thought it's 10 to the minus 10?
10 to the minus 20, 10 to the minus 12, I think, a nanometer.
Thank you.
To a meter. A meter is about a yard. I don't know how many feet are those, but like about this. So 10 to minus 12, thus. Just incredibly tiny, incredibly tiny. Yes.
And getting back to what you said, some people can get used to spiciness or cold. From what you've shown here, it looks like you mentioned it's kind of mechanical in a way. So, for example, I used to be a duck hunter, and I could sit out in the cold weather for hours and be in cold water. And my brother from California was freezing at 60 degrees.
I know.
So how do you take something that's so, it appears to me simple, mechanical, and teach yourself, don't have those things open and close so much?
We
Drugs,
are in
man.
Michigan. Do not open. Well, well, there's a process known as desensitization, in which, how to explain this? So every time that maybe I'm gonna go back to the temperature receptors, right? So those ones. So every time we have capsaicin, they will open and they will have a response. But if we do that over and over and over and over, cells are like, okay. So there are many mechanisms that allow those cells to not respond that much, because if those neurons are firing like crazy, that is not okay. So one of the things that can happen is, imagine that a single cell has 1,000 of this. This is a completely random number that I just invented. If we keep activating them, maybe the cell will not send that many outside. maybe we're going to have 800 instead of 1,000. So then you will not respond as much. So that's a possibility. The responses inside of the cell can also be modulated. So there are certain medications and drugs that throughout the months or years have to be adjusted, like the dose needs to be increased because there's tolerance or resistance. Tolerance is just that. Cells being very smart and saying, you are using these channels too much. So we're going to decrease the response. And yeah, we are quite good at adapting to our environment. So someone in California, 60 degrees, and they're crying. I'm like, you know, they're used to hot temperatures. So we go down there to California, we're going to be crying in, and I say we, I have been here for 8 years, so I consider myself now a Michigander. So I'm really good at, I mean, not as good as you are, but I'm better than my family down in Mexico. They just see the snow and they freak out, and I'm like, come on, it's just 0 Fahrenheit. We're okay.
Assuming that, and I'm making an assumption that the DNA that I have is the same DNA I had in my body when I was born. How is it? What impact does age have on this? Because there are certain foods I used to crave as a youngster. I no longer crave them, nor can I stand them. I used to not be at all subjected to heat and cold, you know, but now as I get a little older, cold has a little bit more impact on me. So
Sure.
What's changing?
So the DNA is exactly the same. It's just your physiological response, this adaptation. So for example, body temperature regulation is not depending just in this. This is the first of many steps. We have hormones regulating that. We have sweat, we have shivering, we have many, many things. And as people age, this is something not uncommon. People get less ability to fight the cold. So then we have a lot of seniors going to Florida and to California and Arizona because, you know, it's warmer. And maybe you don't have to be here shoveling snow. Like I wouldn't like to be here shoveling snow when I retire. I'm going back to Mexico. Yes. Yes.
When you mention the word desensitization, it brings back to me a harrowing episode in 1987 in my medical training, where we had a pediatric patient with an infection that would kill this child if we not treated with a particular antibiotic to which she was allergic. And we did a desensitization, starting with 1/1000th of the dose. Let her get used to that. Then 1/500th of the dose, 1/100th, 1/50th. And finally we could give it, and she did okay.
Yeah.
Hopefully she's still alive. But that, I'm wondering about the parallels between that. That's a chemical desensitization.
Yeah.
You're talking temperature.
You can do chemical, you can do temperature. It works pretty much in every receptor we have. So what you were doing there probably by, because this was an allergic reaction, this was a cellular reaction from the immune system. So you're just teasing a little bit. So there was some, it's like a vaccine. You tease, but then a little bit more, and then a little bit more. This process also happens with people who abuse drugs. That need for more and more happens because the drug, let's think cocaine, doesn't work as well anymore because their neurons desensitize. So to get the same effect, you need more. And it happens also with drugs of prescription. It happens with many things. Vaccines are a way of desensitizing the immune system. You're just like teasing. But when you're overactivating after an extreme activation, then the response will be decreased. So it's quite interesting. We can adapt very well to whatever we are faced with, which is wonderful, right? At the very molecular level, these tiny things it's like basically we're superhumans. We have it's a miracle that we're functioning alive. It is incredible.
Any other questions? Erika, thank you very much
Thank
once
you
again.
all
Well
of you
done.
for your questions.